Hello and welcome back to the nervous system. This is part 3 of my walkthrough of this PowerPoint for my Biology 231 class. Hello class. So if you want to see the action potential drawn out using ions and channels across the membrane, pause this and head over to my drawing tutorial that's specifically about that. We're going to walk through this verbally and then I'm going to show you some pictures. So this should pander to all the folks that aren't super into drawing but are really good at bullet point and sentence-based learning. So, how is this different from a graded potential? Well, for one thing, action potentials travel. So when we say that a neuron conducts an impulse, whoopsie, go back. So when we say that a neuron conducts an impulse, what we mean is the impulse is traveling along the membrane. It's not staying in one place. So adequate stimulus is required, and what we mean by adequate is a stimulus that reaches threshold or above. So it causes the membrane to raise to at least negative 55 millivolts or above that. So sodium enters the cell, causing depolarization. So remember, depolarization in this context means inside of the cell increases in positive charge. So the millivoltage goes from negative more towards the positive. So the threshold potential in this case is negative 55 millivolts, and that is what is required to open voltage-gated sodium channels. So the adequate stimulus part creates a graded potential that's mediated by ligand-gated sodium channels, but you have to have enough ligand to open enough of these to let enough sodium in to have the membrane reach negative 55. So that's the relationship between the stimulus, the threshold, and the voltage-gated channels. So sodium rushes into the cell, causing the cell to pass 0 millivolts and head up to between 30 and 35 millivolts. Now this varies a little bit across neurons, and you'll see anything from between 30 and 40 millivolts reported in various different literature. Don't be too married to the number. It's just in that range. So you'll see it reported as between 30 and 40, so 35 is right in the middle. And it does vary a little bit for different neurons too, but we're not going to really get into why that is. So this is the range between negative 55 and around positive 35. That the voltage-gated sodium channels remain open. After that range, they close. They only have a particular voltage range they like to be open during. However, positive 35 to 40 is the voltage at which voltage-gated potassium gates like to be open. And so they're going to open and let all the potassium that's been stored up inside the neuron during resting potential out. So we have outward diffusion of potassium, and that results in repolarization. And the repolarization goes so far that it undergoes a mild overshoot. So this means it doesn't just go back down to negative 70, it actually dips below that as much as down to negative 90 even. So that's called hyperpolarization because hyper means more than, so this means just more polarized than RMP. Once hyperpolarization has completed, the membrane potential has been restored after that by the sodium-potassium ATPase. So remember, we just let a bunch of sodium inside the cell and then we let a bunch of potassium out, which is the opposite of normal conditions. So what the membrane looks like after hyperpolarization is all the sodium has come in, so there's lots of sodium inside, and then a bunch of potassium is out here now. And that's the opposite of the way it is at rest. So we need somebody to reverse this, kick all this sodium out, let the potassium back in, and that's the sodium-potassium pump. Okay, action potential. If you measure voltage changes over time And I want to stress this is unitless, but this time is in milliseconds, so thousandths of a second. We have a neuron kicking along at negative 70 millivolts, but a local potential is developing across the membrane. And this local potential is due to ligand-gated ion channels. So let's write that. And specifically ligand-gated sodium channels. Threshold is negative 55 millivolts here, and at negative 55 millivolts, voltage-gated sodium channels open and initiate an action potential. So during this part of the curve, we have voltage-gated sodium channels open. The voltage-gated potassium channels are shut here. Opposite is true during repolarization and after hyperpolarization. So on this side of the curve, voltage-gated potassium channels are open. So this area is after hyperpolarization. And then when the potential begins to rise again in this little stretch of membrane, that is when the sodium-potassium pump kicks back on and restores everything to normal. So getting comfortable with looking at a curve like this and labeling the curve with which channels are in charge of which phase is going to help you best prepare for the exam. So a cleaner, neater version of this exact same picture is forthcoming now. So you can see this divides the curve up into bars and tells you which channels are open at which time. So the same way to represent what I just drew you. So we call this a spike, and spikes follow an all or nothing principle. So you either get opening of voltage gates or you don't, which means you get an action potential or you don't. These are non-decremental, so they don't get weaker with distance, and let's add from soma. So the amplitude of this curve, which is its total height, does not diminish from the axon hillock to the axon terminus. And it's also irreversible. So once you trigger an action potential, you cannot stop it. It will zip down to the axon terminal, whether you want it to or not. So you can't initiate an action potential and then, oops, change your mind, you don't want it anymore. That's not possible. So why is it irreversible? Well, the answer to this has to do with the refractory period. So the refractory period refers to the period of time, and it's very, very short. So it's only half a millisecond, which is 1/2 of 1,000th of a second. So absolute refractory period just means that when you're already depolarizing, you can't double depolarize. So you can't make the action potential this much taller, which is what that would mean. So this is already occurring, you can't do it again. The relative refractory period means that it's theoretically possible to re-excite, but because of the channels that are open presently, only an extremely strong stimulus is going to trigger a new action potential here. What this also means is that because we have an absolute and a relative refractory period for each spike, there is a maximum number of spikes that can be on an axon at any given time before they start to run into each other. So what you see here is these increasing local potentials rise and rise and rise, and then they level off because in this part of this particular graph, the maximum number of depolarizations per unit time has been achieved. So this area is maxed out, can't carry any action potentials at a higher rate than it already is. So let's talk about impulse conduction in unmyelinated fibers, and then we'll get to myelin. So unmyelinated means no nodes of Ranvier and no internodes, be they oligodendrocyte feet or Schwann cells. So we don't have the myelin sheath here. And there are plenty of unmyelinated axons. They primarily exist in ganglia and in the retina of the eye and some other places. And they do serve a purpose, and when we get to the autonomic nervous system, you'll see some more of these. But they go slower. So because of the way they are built, the action potentials that are able to move down them happen at a slower rate. So here's how this works. Threshold voltage in the trigger zone begins the impulse, so that's -55 at the axon hillock. And the membrane polarity reverses resting membrane potential. So that causes electrical current to flow between the site of the action potential and adjacent membranes. So let's say I'm letting a bunch of sodium in right here. What that means is in the red chunk of membrane, there's suddenly more sodium than normal. So that sodium is going to diffuse through the axoplasm to areas where there is less sodium. And that serves as the stimulus to excite the next chunk of membrane. So this is next. Why doesn't it cause depolarization behind itself, even though the sodium does indeed flow over here? Well, in this chunk of membrane, voltage-gated potassium channels are open and voltage-gated sodium channels are shut, and this isn't enough voltage-gated sodium channels to reverse the repolarization or after hyperpolarization. So that keeps the action potential moving this direction, not this direction. So the chain reaction proceeds down the axon from the soma towards the telodendra. So now we have an influx of sodium here, and then that sodium diffuses over here. And this one begins to light up, which is what you're seeing in this. So time 1, time 2, time 3, we can see the action potential has progressed toward the end. So this is non-decremental, meaning it doesn't lose amplitude or power, but it's relatively slow. So about 2 meters per second. This is slow in nerve terms. Obviously, 2 meters per second seems pretty fast when we think of our own locomotion, but that's nothing for neurons. So myelin fibers of the same size will move at between 3 and 15 meters per second, depending, with large myelinated axons carrying potentials at 120 meters per second. So in addition to myelination, diameter also increases speed of transmission. So the action potential never reverses due to the refractory period, as we've already established. So what happens if you do have myelin? Why is it faster? Well, the travel of the nerve impulse along myelinated fibers doesn't rely on the diffusion and excitation of the immediate next membrane, but rather due to rapid axoplasmic diffusion of sodium between the nodes, the action potential is going to jump from node to node. And one of the reasons for this is that the voltage-gated channels, because they're confined to the nodes and the nodes only, are extremely numerous in those nodes, meaning that when they snap open, so much sodium is allowed to enter that little tiny chunk of axoplasm, and it very rapidly diffuses to neighboring zones of the axon. So I'll show you a picture of this in a minute. But voltage-gated channels occupy up to 12,000 of them per micron squared at the nodes of Ranvier. That's a lot. So they're very densely packed in there. So the speed of nerve conduction is dependent on, again, the presence of myelin and the diameter of the nerve. So more myelin and bigger diameter equals faster conduction. The reason it's called saltatory conduction is because saltatory means movement by jumping. So because the potential jumps from node to node, we call it saltatory. So here's what this looks like. Here we have a picture and then a time course image So this is showing you all the little sodiums. So these are sodiums, and they are only able to enter the axoplasm at the node, where there are 12,000 gated channels per micron. So once we have an influx of sodium here, notice now there's a large bolus of sodium here, and it's confined to the axoplasm of a tube. So it's going to really rapidly diffuse this way and this way, but it can't do anything because this area of the axon is refractory, so that's ineffectual. But up here, this is resting membrane potential zone. So the rapid diffusion of sodium over to here is going to serve as the stimulus for these voltage-gated channels to open. And the same process is going to repeat itself over here, such that the next chunk to depolarize is this one. So that's what you're seeing in these bottom images. So this is repolarizing, this is depolarizing, and this is resting. So at time 0, you have the action potential here, refractory area here, and yet to be depolarized here, and then the rapid diffusion of sodium from the action potentialing area travels to here, which serves as the signal for sodium influx into this chunk of membrane. And the same thing happens at time 2. So this is time sub 1, this is time sub 2. So you can see the red area, instead of proceeding underneath the Schwann cell to the next zone, just depolarizes, and then the next area is, boom, over here all of a sudden. So that's what we mean by jumping. The area that's depolarizing jumps from node to node. Okay, so types of synapses. So now we're getting into our discussion of what happens when the action potential reaches the end of the axon. So it would be pointless to produce action potentials if they didn't do something, and we know from the neuromuscular junction that one of the things that is possible for them to do is to excite a muscle cell. But in the nervous system, nerves are exciting usually other nerve cells. So let's look at the 2 ways they do it. Way number 1 is electrical synapses. So this is an electrical synapse. And as you can see, 2 cells are joined by tight junctions, which prevent things from going between the cells. And in the center, there is a plaque of gap junctions, and these are open tubes such that this cell can exchange electrical charge with this cell. So in the nervous system, these are fairly rare. The place that you might see them, in fact, you have already seen them, is in cardiac muscle tissue, because heart muscles have intercalated discs, which are tight junctions, desmosomes, and gap junctions. So that's what allows your heart to rhythmically beat as a muscular unit instead of the individual muscle cells beating by themselves. In a chemical synapse, which is what we're going to focus on in our class, there is a presynaptic cell, and that culminates in a synaptic knob. And those release neurotransmitters, which are chemicals that diffuse across a small gap called the synaptic cleft to a postsynaptic cell, whatever that may be. So we have an action potential, which is an electrical signal, coming down here and briefly being changed into a chemical signal, and then due to the action of receptor molecules, this cell may experience another action potential, or not, depends. So this picture, we are showing you a scanning electron micrograph of a synaptic knob. The asterisk is in the synaptic cleft. And this area on the postsynaptic cell that is really dark has lots of neurotransmitter receptors. You can also see very clearly, see these little dark circles? Those are all vesicles full of neurotransmitter. And this is a mitochondrion. So neurotransmitter is released from these synaptic vesicles when the vesicles fuse with the membrane. And you can actually see all these little black specks in here. Those are little bits of neurotransmitter in the cleft. Isn't it cool that we can actually see a picture of this? So we have trained you to think about axons synapsing on muscle cells, but we haven't really discussed how they synapse on nerve cells. And it turns out the arrangement varies. So all of these arrangements start with axo, because it refers to an axon synapsing with something, but they vary in their position. So an axodendritic synapse refers to a place where an axon is synapsing upon a dendrite. So here. Axosomatic refers to axons converging upon and synapsing on a neurosoma. And axoaxonic means axon is synapsing on the axon of another neuron. So in the case of axoaxonic synapses, you've actually seen one of these before. So there are GABAergic neurons. GABA is gamma-aminobutyric acid. You'll see this again later. Don't worry if you're like, what is that? I don't know it. So there are GABAergic neurons, and these guys are going to inhibit skeletal muscle motor neurons. And this helps prevent the overexcitement of skeletal muscle motor neurons. If you have tetanus, GABA is not released from these. And that causes overexcitement of the motor neurons, and that equals tetanus complete tetanus of skeletal muscles beyond your control, which we call rigid paralysis. So you've already seen the consequences of these. Axoaxonic relationships are really important. Okay, so chemical synapse structure. This picture should look familiar to you. So we've got a axon, and we have a synaptic knob, which is the broad widened end. And then you can see that there are vesicles full of neurotransmitter here fusing with the foot to release the neurotransmitter into the synaptic cleft. And then presumably these little red guys are receptors. So we have lots and lots of neurotransmitters. So because this isn't strictly a neurobiology class, and because we don't have the time to cover the action and chemistry of every single neurotransmitter, we're just going to sort of give you a broad tour. So one that you already know is acetylcholine. So that is one that you form by gluing an acetic acid to a choline molecule. And these can be both excitatory and inhibitory. So how can one chemical both excite and inhibit? Well, it depends on receptor types. Different receptors have different actions, and we'll see this again later. Amines are synthesized by replacing the carboxyl group in amino acids with another functional group. So monoamines are one, catecholamines are another group, and indolamines are serotonin and melatonin. So this one regulates mood and vasomotor tone, and this one regulates circadian rhythms and sleep, and it may be cytoprotective, meaning anti-cancer. These ones are fight or flight. And this one is part of the brain's reward center. So it's a chemical gift that your brain gives to you for doing something it considers good. Amino acids are among the most common neurotransmitter in CNS. So these are stored in vesicles and used as neurotransmitter. So glutamate is one, and GABA, as I've mentioned, is another one. So it turns out most of your special sensory receptor cells, so like retinal cells, and let's see, olfactory cells, and hair cells. Most of these guys release glutamate when they're excited, so when a photon hits one of your rods or cones, it blurps out some glutamate that excites the bipolar cell nearby. GABA is primarily inhibitory. Other small molecule transmitters are often derived from amino acids, or are a product of some other kind of metabolism. So nitric oxide, NO, is one. Postsynaptic cells can also signal the presynaptic neuron using small molecules that are resulting from the metabolism of that postsynaptic cell. So it's a way for postsynaptic cells to feed back on a neural pathway by chemical signals rather than by using an axon or a collateral branch of an axon, which is pretty neat. So here are some pictures of the classes of neurotransmitter I just showed you. So here is acetylcholine. This is what GABA looks like. This is what glycine looks like. Here's aspartic acid. Here's glutamic acid. Another way to say glutamic and aspartic acid are glutamate and aspartate. The monoamines have ring structures, either one or two. So the catecholamines have one ring. And the indolamines are not limited to a specific ring scheme. They have one or two. Melatonin happens to have two. Here's histamine. This makes you itchy. That has one. Neuropeptides are neurotransmitters that are chains of amino acids. And in this case, the amino acids are bound by peptide bonds. So that's why we call them neuropeptides. So enkephalin is important in pain blocking. So it's an endogenous, meaning one that you make your own pain blocker. It's the opposite of substance P. This is P for pain, so pain inducer. This is a neurotransmitter that is specific to pain. Cholecystokinin has a role in digestion, and beta endorphin is one of those endorphins, so it's a neurotransmitter that helps to do things like ignore pain or feel good or euphoric during stress or activity. So it's just sort of a rogues gallery of different neuropeptides. There are many others. I'm just showing you what they look like. So here are some more. Chains of 2 to 40 amino acids, so they can be as small as a dipeptide and as long as 40. And these are stored in the axon terminal in larger secretory granules. And that's because instead of just being a small molecule, they are long chains and they have to be wadded up. So these are called dense core vesicles because they've got a lot packed into them. These are good at acting at lower concentrations and they tend to have longer lasting effects. Some are released from non-neural tissue, like the gut-brain peptides that cause food cravings. Some function as hormones, and they can modify the actions of other neurotransmitters. So, for example, epinephrine and norepinephrine are actually considered to be neurohormones because they have a role in both the nervous system and the endocrine system. Okay, so I mentioned that we didn't really have time to get into all the kinds of synapses, so we're just going to sort of show you a few choice ones that are really cool. So we're going to show you an excitatory cholinergic synapse, an inhibitory GABAergic synapse, and an excitatory adrenergic synapse. So these two are mediated by ligand-gated ion channels. And this one uses second messenger stuff. So kinases and cyclic AMP and all that, you'll see a picture of it later. There is a slight delay in chemical synapses, and that's the time it takes for the neurotransmitter to diffuse across the synaptic cleft. So time from arrival of nerve signal at the synapse to the start of the action potential in the postsynaptic cell. So all the time it takes for this stuff to happen. So binding, release, binding, excitation. So here's one that you recognize already because we have looked at these in the form of the neuromuscular junction. So what happens when the action potential gets down here? Voltage-gated calcium channels open and let calcium into the cell. Due to a complex signaling cascade that we won't really get into, calcium influx causes release of neurotransmitter, so fusion of the vesicle with the membrane in exocytosis. Acetylcholine receptors trigger the opening of potassium channels, so that lets potassium into the cell, and that produces a local graded potential. When, and I'm going to cross this out and say if, because if you don't have enough neurotransmitter, it may not reach negative 55. If the local potential reaches negative 55, an action potential in the postsynaptic cell will occur. All right, inhibitory GABAergic synapses. So I mentioned that this is also based on ligand-gated ion channels, and it's true. So GABA stands for gamma, so this is the Greek character gamma. Gamma-aminobutyric acid, so GABA. So in this picture, GABA is pink triangles, and when GABA binds to its receptor is also a chloride channel. And so chloride is an anion, so if chloride goes inside, that's going to make the inside of the cell more negative, and that hyperpolarizes it. So hyperpolarization occurs when you allow the inside of the cell to become more electronegative than usual. So now it's harder for the postsynaptic neuron to reach threshold. So if I were to draw that, so this is time, and this is millivolts, and let's say this is negative 70. So my cell is kicking along at negative 70, and then GABA is released and is acting on the cell at this point. You're going to get a little blip in the downward direction that is going to push the membrane further away from threshold. So this is called an inhibitory postsynaptic potential. So adrenergic synapses are excitatory, and this makes sense, right? Because adrenaline, or norepinephrine, is one of the neurohormones of arousal, and I mean physiological arousal, so getting your body into a ready state to do a thing, either fight a bear, or punch someone in the face, or run away. So the neurotransmitter is norepinephrine, but notice binding of norepinephrine itself doesn't open the receptor and let something in. That happens later. So the receptor is an integral membrane protein, but not a channel. Associated with a G protein. And G proteins are intracellular, so within cells, proteins that are capable of activating signaling cascades that have downstream effects of great number and variety. So in this case, the G protein activates adenylate cyclase, which breaks 2 inorganic phosphate off of ATP to form cyclic adenosine monophosphate, which means adenosine and only 1 phosphate. So now we've taken this molecule from energy to signaling. So ATP is really useful. Depending on what you do to it, you can make all kinds of stuff. So cyclic AMP has multiple effects. It can synthesize new enzymes, activate existing enzymes, open ligand gates, and even produce a postsynaptic potential. And in the next slide, you'll see it do those things. So let's take a look. So here we have neurotransmitter. Let's say, for the sake of argument, it's norepinephrine. And it's going to bind to its binding site on the G protein coupled receptor. So let's write norepinephrine on here, just for funsies. So here's a G protein. And when this binds to this, there's a conformational change, so a shift in the shape of the neurotransmitter receptor that causes the G protein to activate adenyl cyclase, and that's going to rip those 2 inorganic phosphates off of ATP. So let's draw that because they haven't really completed their chemical reaction into cyclic AMP, and that's going to activate a protein kinase. Remember, kinases are enzymes that phosphorylate. And phosphorylate means add an inorganic phosphate. So this is going to phosphorylate this channel. So let's put an inorganic phosphate on there. And that's going to open this channel and let sodium in. So this is the excitatory piece of the puzzle. So it has the same outcome, let sodium in, but it does so indirectly. So the receptor itself is not a channel. I hope that makes sense. All right, so moving on, we talked about both excitatory and inhibitory neurotransmitter and receptor relationships. So now that we've sort of introduced you to those, let's go ahead and look at the specifics, because most neurons are innervated by more than one other neuron, and that means that neurons can do things like process information, so make group decisions, store information, recall it, and make decisions based on the recall of the stored information, which is all really cool. So anytime you have a thought or a feel, you're doing some of this. So this is based on postsynaptic potentials produced by neurotransmitters. So neurotransmitters are therefore very important. Sidebar, because it's always more interesting when we can apply this to things. Certain drugs that cause massive efflux of serotonin or dopamine from neurons cause people to feel super duper good. So drugs like MDMA can do this. So massive exocytosis. If you combine MDMA in high doses with certain antidepressant drugs, like, let's see, MAOIs, for example, you can end up with something called serotonin syndrome. And this only happens if you do this a lot, but serotonin syndrome means you can no longer make and have run out of serotonin. And if you run out of serotonin, because serotonin is so important for not only mood, but other things like the ability to clot your blood, etc., having something called serotonin syndrome, which is a profound lack of serotonin, can be really dangerous. So generally speaking, messing around with your neurotransmitters is a super bad idea, and if you must do it, say to treat mental illness, you should do it under the guidance of somebody that knows more than you about neurotransmitters. All right, sidebar over. So excitatory postsynaptic potentials push a neuron closer to threshold. So here we have negative 55 millivolts. We know that's threshold, and negative 70. So if I'm making this neuron's potential inch towards threshold, that means it's more likely to depolarize. The converse is true of inhibitory postsynaptic potentials, and this should say I. That was a copy-paste error. So inhibitory postsynaptic potentials, we have the opposite case, where the inside of the cell is made more electronegative, so we're pushing it further away from threshold, so this is a larger distance, and that means it's less likely to depolarize. So that brings us to the concept of summation, which is where multiple neurons can talk to another neuron differently depending on what outcome is required, or you can achieve the same outcome in two different ways, sometimes more than two. So net postsynaptic potentials in the trigger zone depend on whether the neuron, or determine whether the neuron fires. So a typical excitatory postsynaptic potential has a voltage of about 0.5 millivolts, and it only lasts for about 20 milliseconds. So you would need about 30 excitatory postsynaptic potentials within this 20 milliseconds, not after, to reach summation. So this gives you a minimum rate of excitation. So in temporal summation, temporal means time, a single synapse, this one, receives many excitatory postsynaptic potentials in a short period of time. So if we count these, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, all within a very short amount of time. Note that mirrors very closely or looks a lot like the outcome here. Spatial summation achieves the same goal. Look, the firing looks the same. But in this case, the 30 EPSPs required to reach threshold within that 20 milliseconds is not the sole task of one neuron, but rather a shared task by several presynaptic neurons, so they can diffuse the work across many. Outcome is the same. So this is a way you can set up different neural networks to do different things. So neural circuits can be sorted according to shape and information transmission. So when I say information transmission, I mean, is the input the same as the output, or does it differ in some way? So in a diverging circuit, one cell, so the signal begins here, synapses on many others, and each of those synapse on many others. So this is a way to amplify a signal or boost it. Converging circuit is the opposite. So we have 1, 2, 3, 4, 5, 6 neurons here, and the output neuron, there's only one. So we've got a reduction in signal neurons by 6 by the time we get to the output one. So this is a way to dampen or reduce signal. You can set up some circuits kind of weird to do specific tasks. So reverberating circuits, there's a collateral branch of an axon. And so the starter neuron is up here, and it's going to excite number 2 and number 3. And number 3 is going to excite not only number 4, but also it's going to re-excite number 1, causing this to start over again. So this is good when you want to prolong a signal or repeat it, which is the reverberation component. So when you yell an echo in a canyon and the sound waves that you made with your mouth bounce off the walls, that's the initial signal, which is like echo if you're boring or lame, or potato if you're cooler and into vegetables, bouncing off the walls so that you get potato, 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 potato. That's reverberation, and this is the same concept. So initial signal, potato, and then potato, 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 over and over again. That was really just an excuse for me to yell potato, but I hope that made sense to you. Okay, so parallel after discharge circuits. So parallel means there are several different lines of communication from one neuron to the output neuron available, and they run in parallel to each other, so parallel lines. So the input neuron stimulates several pathways which stimulate the output neuron to go on firing for a longer time after the input has truly stopped. So let's say I fire this guy off at time 0, and depending on how these neurons are tuned, the impulse on this leg of the journey is going to arrive first, and that's because, remember, there's that 0.5 millisecond delay, so we have 0.5 milliseconds here before this one is stimulated. So a total of 1 millisecond after this one fires, this one is going to go off. If we count here, so 1 millisecond, 1.5 milliseconds. So this one is going to be re-excited again after an additional 1.5 milliseconds, and then 0.5, 1, 1.5, 2, 2.5. 2.5 milliseconds after the initial signal, the output neuron is going to be re-excited again. So that's how it creates that longer time after input has truly stopped thing. All right, and we have reached 44 minutes and 51 seconds. The next segment of the PowerPoint sort of segues into neural disorders and higher order neural stuff. So I'm going to pause here, and we will pick up next time at disorders of the nervous system cells. So I will see you for the next video. Thank you for your attention.